started making blood vessels. And this is one of the examples of such a blood vessel that was cultured in basically in a sort of very primitive bioreactor. So I always thought of myself as somebody who liked to study things and like to ask questions and do experiments, and indeed that's still kind of part of me. But then this guy came along, Willem van Elen, who at that time was already 86. And he said, we have to use those technologies to start making meat. And he actually coerced a number of investigators from Utrecht, Eindhoven, and Amsterdam, sorry, to get a grant from the Dutch government to start working on meat. And halfway that project, I kind of fell in for a colleague that fell ill. And I thought, this is a great idea. Let's move on with this. And so part of me, I guess, this is not my kind of showing that I cannot make slides, but kind of most of us, I think, want to mean something in the world, want to improve the world, and however small that is. So I had that, I guess, from the very beginning, and I thought, well, if I do this, making meat, that could actually have a much bigger impact than the medical stuff that I have been doing so far. And while we were doing that project, and Peter Verstraeten was actually one of the first, he actually wrote that project together with the researchers. And while I was working on that project, it always had some level of public attention, news journals, that sort of thing. And once I was sitting in a train very similar to this one from The Hague to Maastricht, and I was called by a reporter from the Sunday Times. And I had no idea what the Sunday Times was, but apparently well, I knew it was a newspaper, but that was it. And so she said, can I talk to you? Sure. About cultivated meat? Sure. So we had a discussion about half an hour. I had nothing else to do. I was sitting in that train. I was kind of tired from a meeting in The Hague. And she covered that story on the front page of the Sunday Times and said it all over the world. So that was quite unexpected. But that kind of launched me as a media person, I guess, being vocal about cultivated meat. And that led me to the first funding that I got from Sergey Brin. This is the only picture I have of Sergey Brin where I'm also on it. And here he is mostly interested in the Google Glass that I had on my face. This is, by the way, in the backyard of the vegan daycare of his son in Los Altos. That's where he holds office most of the time. Anyway, he said, okay, I need to do something about animal welfare. Let's think about cultivated meat. And he sent some of his disciples around the world to see who was doing this. And there were, at that time, only like 3 or 4 people doing this. So they called a couple and they ended up with me. The guy, one of his disciples, now a good friend of mine, came over to the Netherlands, said, what do you want to do with this? And I said, well, you know, I'm kind of frustrated because the government program had ended and I have no money to do what I want to do, making a sausage from a pig, present it to the world while the pig is kind of honking around on the stage. And that kind of captured their attention and said, okay, Sergey Brin is going to fund this. Fine, excellent. So he, in the end, put like 3 million in it, which for him obviously is nothing. If he waits 50 milliseconds and the stock of Google goes up, he has recovered that. But for us, it was a major chance to make this happen. And that basically led to the famous presentation of the hamburger in London. I mentioned to everybody this little thing cost $330,000. And my friend, who managed the affairs of Sergey, or at least his investments, said, oh yeah, we have to make 3 of them. I said, well, then I need to have 3 times as much money, no problem. So we made those, and we never really anticipated that, but this was the start of the entire industry, which in retrospect, I'm extremely happy with. But we never intended it. This presentation was more or less born out of frustration that we couldn't get public funding for this endeavor. So I don't need to tell you that actually at that time it was known to me why I actually was doing this. First of all, it was, of course, the kick of it, making that meat with the technology that we already had. But then gradually I realized that this was a major societal problem, much bigger than I thought, and that I could make, or we could make, a big impact on the environmental sort of consequences of large-scale animal agriculture. So here are a couple of those numbers. You probably know them. It's actually a major impact on our planetary resources and greenhouse gas emissions. So the why is actually quite clear and becomes clearer almost every day. So we can all become vegetarians, and I'm the first one to realize and tell everybody that you don't need animal proteins, but we love them. So what are you going to do? And the technology to make muscle from satellite cells had been around for at least 15 years or so. When I was at Harvard, exposed to Herman Vandenberg, who made these organoids of skeletal muscle. He was at Brown University at that time. Maybe he still is. He's retired, of course. But and he was making them as sort of models for pharmaceutical trials. So I was aware of that technology. And then when I entered that project that was run in the Netherlands, I said, okay, we can use this technology. We can use satellite cells. We don't need to have embryonic stem cells from the cow and the pig, which was another arm of that project. We can actually use satellite cells and do this. So the two PhD students that I took over were very happy because finally they had cells to work with, and we started to make those organoids. And that has led to what currently the process for us, at least, is. We take a biopsy from a cow. In our case, these are Limousin cows that are 3 kilometers away from the university. I think you guys are doing very similar things. We isolate the cells, separate the muscle cells from the fat cells, let them proliferate in bioreactors, and then let them make tissue, muscle tissue and fat tissue. This is becoming increasingly important because there are a couple of players now in the field who do not really differentiate the tissue and use cells as an ingredient in a plant-based substrate, which is, I think, in my personal opinion, not the way to go. So I strongly believe that we need to actually make tissues out of this, both muscle and fat, to get the nutritional value and the qualities of meat that people actually would like. So of course, if you do this, it's you have to work on a lot of things to make cell culture efficient, to make it cost effective, and to make it scalable. And there are a lot of optimizations that need to be done on a lot of different areas. And over the years, we have worked on that. By the way, when I said in 2013, when I presented this in 2013, Sergey said, "Oh, 2 years from now it will be on the market." I said, "Oh, I don't think so," but he was quite confident of that. So what we do is we take those muscle biopsies and we sort them with FACS. We have now a couple of really nice criteria to very specifically sort the satellite cells that we want. It's still somewhat of a heterogeneous population, but it's getting more homogeneous every time. And then we realized that some of those populations that you identify by FACS are actually, and here we use 2 markers, just CD29 and CD56, that one of those populations is really good at making satellite cells, the other not so good. And it turns out that the one that is not so good, fortunately, is a cell that makes very good fat tissue. So we were kind of lucky there that we used these 2 populations from one biopsy to make both the muscle and the fat. if you look at it a little bit more closely and you do single cell sequencing, then you find that indeed the satellite cell population here in the lower in purple is a reasonably homogeneous population, but there are still a couple of markers or cells that are out there that are somewhat different. And the population that make fat cells here in the middle with the blue actually is a somewhat heterogeneous population. So we use these two now in our process to make both muscle and fat. And here is just to show, this is a publication by a Chinese postdoc who's now back in China and who did his PhD in my lab. And he introduced the CD29 and the CD56 as those markers. And you indeed find that if you do that, PEC7 positivity as a hallmark of satellite cells is a lot higher. He also went on to show that you can increase with small molecules kind of the stemness of these cells, if you like. Currently, we're using other markers as well, such as alpha-7, one of the integrins, which is very, very specific for the satellite cell that we want to have. When you start doing this, not as a scientist, but in a company, you have to make sure that this is a robust system, that if you take samples from cow 1, they're not very different from cow 2 and 3 and 4. So here is a number of very boring growth curves where we have here somewhat like 20 isolations from different cows, and you see that they're all kind of similar. They grow pretty much at the same rate. So that's helpful if you want to start an industry. Of course, you know that these are adult stem cells, and they at some point give up. They actually continue proliferating. At least half of the isolations that we get, they keep on proliferating pretty much forever. So they are immortalized, if you like, but they do stop differentiating after a certain number of doublings. And also that is actually somewhat variable from isolation to isolation. Oops. So currently we are at 32 doublings where they still differentiate nicely. So if you do that math, from half a gram of tissue, you can get kind of 2,000 kilos of meat. That, by the way, would reduce the number of cows in the world 160-fold, which is appreciable. Of course, you have to scale up production, and for that we use bioreactors. You cannot grow them in 2D. That's almost it. We did that, by the way, for the hamburger. I think there are photos on the internet where we have an entire counter full of 10-layer cell factories that we use to make that hamburger. So that was not particularly sustainable. Lots of plastic in that process. Currently, we obviously are using bioreactors, and there are two ways of doing this. You can grow them on microcarriers, which works very well. The commercially available microcarriers, the cells adhere to them, and they grow nicely, and you can harvest them. It's a little bit cumbersome, but it's doable. And we now are up to 50-liter scales where we can do this quite effectively. So a process would look like this, where you start with a seed train with small samples from a biobank. Then you gradually grow them up in larger kind of tanks, and then eventually, in these 1,000 or even larger tanks. Some people in the industry say they can go to 300,000 liters. We have not seen that yet. I think the biggest we have seen so far is realistically is 500 liters. So the thing with the microcarriers is that you have to separate the cells, of course, unless they are edible, which they currently, at least the commercial ones, are not. And then you develop the fat and the muscle tissue. I mentioned the microcarriers, but we can actually also grow the cells in aggregates, just like you do with embryonic stem cells and with induced pluripotent stem cells. You can grow them in aggregates, which has a lot of advantages because you can increase the cell density in the bioreactor a lot more. The problem with every kind of advantage comes with a problem, it seems, in this area. So Here, the issue really is size control of those aggregates. When they become too big, you can all imagine the center actually starts dying because of lack of oxygen and nutrients. So size control here is one of the biggest challenges and the nice challenges that we are currently working on. So if you with our current kind of values, if you do the math and you want to replace, let's say, a farm in the Netherlands with 250 cows that produces like 35 tons of meat per year, you can actually replace that by 4 of these 1,000-liter bioreactors. Now, this is only proliferation. I have not talked about the differentiation yet. That actually comes in addition to this particular process. But this seems kind of doable. But if you think about it, just to replace beef in the entire world, you need to increase the fermentation capacity by at least a factor 1.5. And all these numbers are still somewhat not very certain. They have a relatively high level of uncertainty. We could easily be a factor 10 off. You have to keep that in mind with everything everybody says. Okay, that's the scaling up. Can you make actually tissue that resembles real meat, or is this a kind of poor derivative? Of course, if you don't differentiate the muscle or the fat, then by definition, it's a poor derivative. If you start differentiating this, then you might be able to get there. So these are the muscle fibers that we typically make. And you see that they are nicely kind of myotubes, and they are nicely merged into large fibers. They are nicely axial. You find also cross striations as a measure of, that there are actually sarcomeres there, but it still doesn't really look like a skeletal muscle that you take from a cow and put right under the microscope. Some of the organization and some of the protein is still missing. So we have worked a lot on the differentiation method for this, and typically, as you know, if you differentiate muscle, you start to you use, in the olden days, you use serum to cultivate them. If you withdraw the serum and withdraw kind of the growth factors in that serum, they start to differentiate. That's the classic way of making muscle cells from satellite cells. So we obviously we did that in the beginning too, when we were still using serum. But when we moved away from serum, we had to figure out, well, what is then the differentiation medium going to be? And so what Toby in the lab and in the company did, he did RNA sequencing of all those differentiation steps, looked at which, when you withdraw serum, look at which kind of molecules start to, and especially receptors, start to be upregulated. And here, so you have the oxytocin receptor, and you have some G-coupled receptor, and an LPA receptor, and IGF, of course, and a cholinergic receptor. So he started to use ligands for all these receptors to see if you can improve that serum-free differentiation medium. Okay, so this is one of those results where he identified a couple of those components that really worked. Here you have, here you have myosin. This is with serum withdrawal. Quite nice bands of myosin. This is with the serum-free differentiation medium. Not quite as good as the serum withdrawal. Desmin is okay. Myogenin is slightly better. Tropomyosin, and we have looked at a couple of others. So here you see this is actually quite an old picture where, let's say, 50 or 60% of the cells merge. This is in 2D. And currently we have improved this to more than 90% of the cells actually participating in one wave into merging into myotubes in 2D. We also make 3D structures, which we call BAMs, bioartificial muscle. This is actually in tissue engineering country is a kind of a familiar term. And here you see the protein content of that muscle. This is minced meat from a regular supplier in the Netherlands somewhere. And you see that in protein content, we are quite in the same range. We see that myosin and actin are still somewhat lower, about half of what is in the original tissue. And myoglobin is also half, which is kind of at the range of pork. This is the myoglobin band that we get from some of those BAMs, and yeah, if you compare that with the regular meat, then that's somewhat similar to pork. In 2013, we didn't make fat tissue yet. And as I mentioned, from that biopsy, we can actually get also fat precursors, FAPs, fibroadipogenic precursors. And they are IgA7 or alpha-7 negative. And they're also, they're sometimes CD56 positive. And they are IgA5 positive, and there are a couple of other markers. PDGF are positive, so we have selection criteria for those cells. And currently, we actually have 90% of the cells that we get out of them differentiate into fat cells. And they can become really big, like here, really monolobular fat cells, as very similar. They're not quite as big. A fat cell that you take from a cow is typically somewhere between 150 and 200 micron in diameter, which is all fat. Ours are about 50 microns in diameter. Again, also all fat. What is interesting is the fat composition, the triglyceride composition of these fat tissues that we make. Here you see it's a busy slide, but it's actually a time scale here. This is day 0, day 7, day 21, day 28. And the color coding refers to fatty acids from 16 to 22 carbon atoms with a level of saturation in there. And you see that in the beginning, in the first 7 days, it's quite unstable, the fatty acid composition. But over time, it actually stabilizes. And if you then compare this on the right too, that's fat that you take from a cow, same fat analysis, and on the left, the 2 samples that we made, and you see that fatty acid composition is very, very, very similar, which was a lucky shot, by the way. We just did whatever we thought was necessary to differentiate them, and it turns out that their fatty acid composition is quite similar, and the taste is also quite similar. Okay, so then there are a couple of other things that you need to worry about when you start doing this. Obviously, you want to make everything animal component free. So we had to get rid of the serum. That hamburger in 2013 still had serum, and also a couple of other animal components in the gel that you make that you use to create that organoid, that muscle organoid. So we had to replace all of this. I will not cover everything because that for the sake of time. But of course, you know, serum-free medium, and I know you guys with what's called beefy, you have done the same. We have done many of these screens and optimized the medium, and we're still optimizing the medium for almost every day to make it cheaper, to make it simpler, and to make it more effective. But here you see that indeed, as you also have shown, that you can generate or assemble a medium that is as proliferative as serum. That's not the problem. The problem really is, can you do this under all conditions with microcarrier growth, with aggregate growth? Do they still differentiate? So we find that we time and time again have to go back and say, okay, we need to go through all these steps. If we change something somewhere down the road, something might change, and we have to go back and iterate. And that's what we are doing all the time. So the evaluation process becomes more and more complex. One particularly interesting aspect of this whole, I find also scientifically interesting, is that is kind of the metabolics of the cells in cell culture. So this is a slide, it's already an older one, but we have done this now many times, where we basically over time look what is the amino acid usage of these cells. On the left, you have the essential amino acids. On the right, you have the non-essential amino acids. And you would think that with cell growth, you actually deplete those amino acids because they have to make proteins. And you indeed see that. So here for valine, for instance, and threonine and isoleucine, you see real depletion. This, by the way, is after 7 days. So it's quite already a long culture without any medium exchange. But you see also that some of those amino acids actually don't drop, even though the essential ones don't drop in level. So typically, after 3 days, you exchange the medium, or maybe you are a little bit more prudent, and after 7 or 8 days. But still, if after 7 days you throw away the medium, you throw away a lot of resources. So we obviously don't want to do that. So we are more and more working on recycling. We can now up to 1 million cells per milliliter in a bioreactor. We don't need to change medium at all. And we now start to supplement some of those amino acids that are depleted. Of course, I hear you thinking, how about ammonia and lactate? Lactate typically is not a problem for these cells. It's, by the way, there is some difference between the fat cells and the muscle cells. Ammonia, of course, is, especially for the fat cells. And but we now have created conditions in that culture where ammonia actually decreases over time and not increases over time. So this is all possible. And then you can start to replace some of the amino acids. You may be familiar with this study. Eventually, we're doing this for with the purpose as an environmental benefit. This is one of the recent studies from CE Delft, where they looked at the environmental impact of this whole thing. And this is a compound score composed of global warming, land use, water consumption, fine particle matter, toxicity, antibiotics, and all those type of things. And then you see, that's not a surprise, everybody finds that beef and dairy are the worst. Pork and chicken are a lot better, and on the left is vegetables or plant-based proteins. And there are two cultured meat entries in here, conventional and sustainable. And the difference between these two is essentially where you get your energy from. This is presumably an energy-intensive process. There's still also a lot of assumptions here, but let's assume for the moment that it's an energy-intensive process. And of course, then it becomes really important where you get your energy from, whether this is from fossil fuel, which is even too ridiculous to think about, or from sustainable sources. And then indeed, these calculations show that you could, again, a lot of assumptions here, that you could have a major environmental benefit, even over chicken and pork. We also know, of course, that this is an expensive enterprise. Cell culture is not cheap. And so a lot of our colleagues in the field say, okay, this is never going to work because it's so expensive. And how do you ever, how are you ever going to make it cheap? Fortunately, our friends of the Good Food Institute have created a couple of reports where they said, okay, if you get a large scale growth factors at low prices, if you remove serum, if you make it yourself, which is a big improvement, then maybe you can get into that range where the medium is becoming cheap. We added to that recycling, and if you do those calculations, currently we could produce it for about 140 euro per kilo, which at the current exchange rate is $150 per kilo, $140 per kilo. So that's, for a tissue engineered product, is exceptionally low. for a meat product, it's still way too high. So how do you get from there to, let's say, 50 cents per kilo? And that's essentially where one of our projects comes in, where we use very raw materials like fodder beets and pea proteins, isolates, to replace some of the components in the medium. And this, I apologize that the x-axis is anonymized. These are protein hydrolysates from different sources, ranging from wheat, soy, cotton, all sorts of different protein sources. And they are hydrolyzed in different ways. The color coding refers to the different ways of hydrolyzation. And here 100% this is in a medium where all the amino acids have been taken out. So if you have 100%, basically the hydrolysate works as a perfect replacer of the amino acids in the medium. And you see that some of those hydrolysates, a lot of them actually don't, but some of those hydrolysates actually work quite nicely in replacing those amino acids. So I think this is the way to go. We are not the only ones. There are a couple of others who have, I've been talking about this for many, many years, but now we have a reasonable amount of data to show that this is actually possible. So of course there is there's always also the sort of consumer acceptance, and this is not particularly my field, but you have to make it your field if you want to be in this business, whether you like it or not, especially if you go public in 2013 with a hamburger like this. And people said, yuck. And nobody is ever going to eat this. And I thought, why not? People eat hot dogs. Why don't they eat this hamburger? So I had to kind of develop an idea why this is different, and it's still there are a couple of components. I won't talk about it too much. It's an interesting field, but there are a couple of components that really make it different from a hot dog. But I won't go into detail. We did one interesting study where a French student in the lab said, okay, I want to do a consumer acceptance study, and I want also to let people taste this. So, well, that's not possible because that's not allowed. And then, okay, then we let them taste a fake cultured hamburger. So that's what we eventually did. And we wanted to test which type of information actually is important for people to affect their acceptance. So we had 193 people, divided them in 3 groups, did a survey on acceptance with a lot of questions, then gave them the information. One was about the societal benefits of this cultivated meat. One was about personal benefits of cultivated meat, that it's healthy and all that stuff, because marketeers always think that consumers are only interested in their personal benefits. So they always think that this is most important to convey. And then we had general information about meat quality and taste and things like that. Then we did, then we repeated the survey, and we let them taste a piece of hamburger, 2 pieces of hamburger, one that was labeled conventional and the other was labeled cultured. They were actually both from exactly the same hamburger, and let them reflect on the taste and the juiciness and all those things. And then we did another questionnaire for acceptance. And this is the outcome. So the color coding is which type of information people got, societal benefit, personal benefit of just high quality. 4 questions: are you in favor of this? Do you want to taste it? Do you want to buy it? And do you want to replace your current meat consumption with cultivated meat consumption? And then we did the questionnaire 3 times: before we gave the info, after we gave the info, and after tasting. And you see that actually, immediately, the color coding really doesn't matter what type of information you give. It's all more or less the same acceptance. What matters is giving information. So before info, lower acceptance; after info and after tasting, higher acceptance. And that's throughout the board. The more information you give, and it doesn't really matter what information, if it's positive, of course, acceptance increases. And we also had, so they tasted it and they found the cultured meat hamburger tasting better than the conventional one, although they were exactly the same. And this is kind of known in the field. If you take a very cheap wine from a jerry can from the supermarket and you pour it in a nice Chateau Margaux bottle from France, it tastes a lot better. All right. Oops. And then this is kind of an interesting aspect. If you, this is just part of the interesting journey, right? You start as a tissue engineer, and you have to do psychological studies. And at some point, you get approached by an imam or by a rabbi. You know, is this kosher or is this halal? And so I ended up, for the first time in my life, publishing in this Journal of Religion and Health. It actually now has about 60 citations, so it's apparently something that people are interested in, but it's about the halal aspects of cultured meat. And spoiler alert, yes, it can be halal if you slaughter the animal, if you don't use blood in the process. What is particularly interesting is that single 2013 presentation led to a current in industry with over 100 companies worldwide. And people always ask me, you know, how do you feel about this? And you know, this is actually the most satisfying thing ever. Yes, there are competitors, but they're also kind of confirming that this is something that can be done and should be done. Of course, this is a novel food. It has to be approved by regulatory officials. In Europe, that's EFSA, that's the European Food Safety Authority. We are personally not working on, or our company is not working on genetic modification. So we don't have that issue. If you do in Europe, it's going to be tough. In other geographies, it might be a little bit easier. As you know, in Singapore, the first product has been approved in 2020, surprisingly with serum. It's a chicken nugget and it's sparsely available in Singapore. So if you want to go there, don't fly because that would not be good for the environment. But you can get it there. So, you know, the take-home message, and this is probably a sort of preaching for the choir, that this is, in my mind, extremely worth pursuing, and there is actually a high likelihood of success. It's still a formidable task to scale it up and to make it commercially viable. Cost effectiveness is part of a lot of endeavors on all the different areas. Technological, recycling, feedstock sourcing, all those type of things need to be in place to make it cost effective, and regulatory approval is required. I actually think that is that's a good thing. We have to show to the public that this is absolutely safe. Of course, we think it's absolutely safe, but you have to show that and get independent confirmation of that. This is a pre-corona snapshot of what the company was at that time. Currently, we are with 160 people and divided over 7 teams. Working on medium development, working on computational science, on engineering, on meat or muscle tissue engineering, fat tissue engineering. So we have divided them over these various, and of course, bioprocess, large-scale production. It's an incredibly motivated group of people. We have never really advertised for any of those positions. They all come through the website, and for every position that we open, we have a tremendous group of talented people, and it's actually hard to choose from them. So this, I think, is also something that this area brings with it, the feeling that, you know, you're working together on a mission. It's not just a job, it's actually working together on a mission, and that brings a energy and a motivation with it that I've never seen before at the university. So with that, I would like to stop, and I hope that there's time enough for questions and that I don't gobbled up all of it. Thanks.
Thank you so much, Professor Post, for that amazing talk. Anyone has any questions, the floor is open.
I guess thank you for your talk, Dr. Post. That was fascinating. So I know that in the Netherlands, you're able to legally do taste testing. I don't know if that's in any lab or just specific labs, but do you think that's going to change the sort of standards or ways that your samples are tested and what research is published? Are you going to be doing less, say, fluorescence imaging, and is it going to be more qualitative? Like what's the outlook given that?
Right. Of course, we don't tell anybody what we obviously taste quite a bit.
But now you can publish.
Right. This is recorded, right? And of course, we tasted in 2013 in London, which was absolutely illegal. And it so happens that an uncle of my wife is a law professor in food. So I asked him before the 2013, what can happen if I get caught with this? And he said, oh, you can get €1,000 fine. Okay, well, given the cost of this entire endeavor, I'm not going to worry about that. But nobody ever paid attention. So fortunately, and you're completely correct that the Parliament a couple of months ago issued a kind of an approval that we can do tasting under specific conditions. And currently the Ministry of Health is defining those conditions. I'm somewhat afraid that it's going to be very similar to a clinical trial, so that it's quite limited actually what you can do in tasting, but we'll have to see. Currently the conditions are not defined yet.
Thanks
Andrew.
for a great talk. I have a question. Super cool hydrolysis amino acid replacement data. That's really, really exciting. I'm just curious, have you done any of the depletion tests with that as well, like you were showing with sort of the individual amino acid depletion in the media over time? Have you looked into that for the hydrolysis?
No, we actually have also not looked at it for the individual amino acids. It's something that we have thought about, of course, and it's not really a priority at the moment. At some point, we will have to know exactly what's in those hydrolysates in terms of we roughly know the amino acid composition, but if you get let's say a pea protein from one day it comes from the pastures here in the Netherlands in the summer, and the other day it comes from South America when it's winter here. Is that the same? Can we actually rely on the composition of these hydrolysates? How sensitive are the cells to changes in amino acid profile of these hydrolysates? And you can go about this in two ways, and indeed do depletion studies, do fractionation. We have done those fractionations, and also look at enrichment, or look at seasonable or geographic variability of the origin of the hydrolysis. So it's a big story. So we're doing this together with one of the largest worldwide merchants of feed products. Feed components, yeah, we are gradually getting some handle on it.
Thank you.
By the way, so far, if we look at some point, if you don't refresh the medium, the cell proliferation goes down. And we know for sure it's not ammonia and it's not lactate. If you then start to supplement the amino acids that are depleted, the result is quite disappointing. So it's not the entire story. We have done, by the way, the same with growth factors, and there it's even more interesting how how growth factors are being consumed and how they seem to be still active while you cannot detect them anymore in the medium. I have theories about that, but it's it's not more than theories.
Thank you for the talk, Professor Post. You mentioned that some companies aren't differentiating their cells, treating the cells more as an ingredient. Do you have any evidence, either nutritionally or taste-wise, that differentiation is necessary?
It's a good question. Evidence, not really. We do well. The evidence that we do have is from tasting panels where, with especially the fat tissue. So if you add animal fat tissue to a plant-based product, you can do with very, very low quantities, much less than plant-based fats. And it definitely adds to the taste. So that's clear. For the protein, it's a little bit more complex. And if you so animal or muscle proteins have a couple of things. First of all, they have the heme iron, which is a valuable source of iron, not the only source of iron, but it's something that's easily digestible by us. You can absorb heme iron without any receptors, whereas plant-based irons actually do need receptors in your gut for absorption. There is the amino acid kind of composition of the whole complex of structural proteins of muscle. And since the structural proteins, the myosin, the actin, tropomyosin, titin, are 75% of all the protein in muscle, the amino acid composition of those proteins actually determine the amino acid composition of the muscle. And then finally, you have the function of the proteins. Actin and myosin, especially in their tight conformation in the sarcomere, glue meat together. So in the cooking behavior and in the water holding capacity, that particular set of proteins and maybe even the particular conformation in a sarcomere are important for the cooking behavior. So of course, also if you if you eventually, this is all kind of a stepping stone towards making full thickness steaks. And for that, for sure, you would need to have that sort of composition of those muscle fibers. So I strongly believe that it's necessary for fat as well as for muscle. What the information that is still missing, and I'm hoping that somebody is going to present that information at some point in time, that if you ferment, if you make myosin and actin and titin and tropomyosin through fermentation, and you make a soup out of it, whether you can actually get that same functionality of those muscle proteins. And I thought that somebody was going to do it, but I haven't seen it yet.
Hi, Dr. Post. Thank you for your talk. I was wondering, if differentiation at scale has been a challenge. Like, for example, I don't know if your approach is this, but like creating the BAMs for muscle at scale. And then I was also just curious about contamination during scale up, if that's been a big deal or just not.
Contamination is a fact of life if you do cell culture, right?
Sorry,
So okay.
can you do the
Oh yeah, sorry, yeah. So so contamination is something that occurs. We are currently tracking this quite intensely to make sure that, if you go from 30 people in a lab to 160 people, it's not 160, it's like 90. Then of course you're going to have more contaminations. Is that proportional to the amount of work that you're doing? Those type of things. So we're very narrowly or very intensely kind of tracking this, and also if there's a contamination, then where it comes from and those type of things. We now have, of course, a quality control officer who swaps the entire area with ATP swaps every 2 days. So yes, it is always a concern. It's not bothering us too much. We want to eradicate it as much as possible, also because we want to get rid of the antibiotics. So we're heavily invested in this. But yeah, it happens from time to time. The other question, scaling up of differentiation, is something I think that is very that's actually where most of the IP, I think, of the companies, at least our company, is, because scaling up in tissues is not an easy endeavor. We have automated this. So our current system is robotized. It still has a very large footprint. So we're currently working also on ways to get the differentiation in a different system where we can actually get it with the same quality and the same degree, but now in a much easier to manufacture way and a much easier way to scale it up. So it's a continuous process. For fat, by the way, it's not an issue. For fat, the issue is that it just takes long. It takes a month for the monolobular fat cells to grow that I showed you. So that takes long, but that actually can be done in a regular stir tank bioreactor. So for that, you don't need anything specific. For the muscle, you do.
Thank you.
Any more questions? No? Okay, no more questions. Thank you so much, Professor Post, for being Cultivate September 2022 speaker. It's an honor to have you. And yeah, that is all.
Yeah. Thanks for having me. And have a great rest of the day.
You too.
Take care. Bye.
Okay, I think if anyone wants to take a 5-minute break, I think we have 5 minutes before we start with the next part of the day. So let's reconvene by like 2:05 in this room. Thanks, everyone. Okay. I don't know, are you?
Okay.
Yes. Yeah.
No, I don't think so.
Internal survey.
And then maybe some Saturday.
Oh, yeah. So. Yeah.
I need more.